Welcome back to the deep dive. If you're involved in pharmaceutical development, and I'm talking about discovery, toxicology, or, clinical regulatory affairs, you already know the ICH M3R2 guideline isn't just a checklist.
It's not at all. It's really the core international blueprint for defining the entire non-clinical safety assessment.
Exactly, and understanding its logic. Well, it can be the difference between a smooth transition to the clinic and a really costly, time-consuming hold. Okay, let's unpack this. This guideline, the full title is Guidance on Non-Clinical Safety Studies for the Conduct of Human Clinical Trials and Marketing Authorization for Pharmaceuticals. But it's basically our global operating system for de-risking new molecules.
And that's really the mission here, isn't it? To get into the operational
Correct.
mechanics of M3R2. The document is so fundamental because its primary goal, beyond just safety, is harmonization.
Harmonization, creating that single standard everyone can agree on.
Right. A mutually acceptable standard among the EU, Japan, and the USA. It defines the current recommendations for all the safety studies that support not just early clinical trials, but also, critically, the final marketing authorization application.
So if that's the goal, harmonization, what are the core objectives M3R2 is really trying to achieve with that?
Well, it's a balancing act. It's a pursuit of efficiency balanced with ethics. You need clarity to facilitate timely trials, of course, but also to promote safe and ethical drug development. And a huge piece of it today is the reduction of animal use.
The three R's.
The three R's, exactly. Reduce, refine, and replace. M3R2 provides the very structure that lets us integrate validated, say, in vitro methods and justify why a particular study might not even be necessary.
So when we talk about the scope here, what's the universe of non-clinical data we're really digging into today?
It's the whole standard package you'd expect. General toxicity, pharmacokinetics, PK, and toxicokinetics, or TK.
Yeah.
Then reproduction, genotoxicity, carcinogenicity.
The big ones.
The big ones. But it also gives essential guidance on the conditional studies, the ones you evaluate case by case. Things like photosafety or immunotoxicity.
Okay, so let's start at the absolute foundation of any program, which is establishing safety margins and that initial dose. The whole point of the non-clinical safety evaluation is to characterize toxic effects, right? Identify target organs.
Understand dose dependence, potential reversibility, all of it. And it all feeds into estimating that first safe starting dose in humans, which is built from the NOAEL.
The NOAEL, the no observed adverse effect level.
Right, the dose where we saw no statistically significant adverse effects. And this brings us straight to one of the most important sections for strategy. High dose selection, that's section 1.5.
And it's so important because we need real scientific rigor here. We have to limit animal doses, to prevent using these excessive, non-relevant amounts of a drug just because we technically can.
Absolutely. So what are the two main boundaries we use to define that maximum dose in a general tox study?
Well, the first is the standard limit dose, right?
The standard limit dose, it's 1,000 milligrams per kilogram per day for both rodents and non-rodents. That's the ceiling, unless you have a very strong scientific reason to go higher.
And the second one, which is often more relevant for potent compounds.
That would be the exposure margin rule. And this rule states that doses giving you a 50-fold margin of exposure. And we usually measure that using group mean AUC of the parent drug.
Area under the curve.
Yep, AUC. So a 50-fold margin compared to the maximum clinical exposure that's generally acceptable as the maximum dose.
And that 50x AUC rule is just crucial. It lets us stop dosing high mass when, solubility issues or formulation problems would otherwise completely derail a study. We use exposure, not just mass.
Exactly. For a high-potency drug, you might hit 50x AUC at just 50 milligram limit. So you stop far, far short of the 1,000 milligram limit. You save animals, you save resources.
But there is a big, complex exception, the one that lets you actually exceed the 1,000 milligram limit. This is for the high-mass clinical drugs. What are the preconditions that trigger this?
Right, this is the 1G-day clinical exception. And it only comes into play if you anticipate a clinical dose over 1 gram a day, and you find that the standard 1,000 mg/kg/day limit dose doesn't even get you a minimal 10-fold exposure margin.
So both conditions have to be met?
Both. If they are, then M3R2 gives you some limited flexibility.
Okay, so we've met the threshold. We need to go over 1,000 mg/kg to get our safety margin. What are the three simultaneous limits that are placed on that new maximum dose?
So once you cross that threshold, the dose you pick has to be the lowest of three things. First, a 10-fold exposure margin. Second, an absolute max dose of 2,000 mg/kg. Or third, the maximum feasible dose, the MFD.
Which is just what you can physically get into the animal.
Right, was dictated by formulation volume or tolerability.
Yeah.
This whole structure ensures you only push to 2,000 mg/kg if it's achievable and necessary to hit that minimum 10x margin for that very specific scenario.
That tiered logic is so important to get. You don't just get to go to 2,000. It's just a ceiling.
A crucial distinction. And we also have to remember the U.S. Phase 3 requirements. If you rely on that 50-fold exposure margin as your limit dose, meaning you stopped because you hit 50x AUC, not because you saw toxicity,
Right.
then you generally have to identify dose-limiting toxicity in at least one species. It's a mechanism to make sure the risk profile is really, really well characterized before you have widespread human exposure.
Okay, let's shift from dose selection over to duration matching. How long do our animal studies actually need to run? This is all about repeated dose toxicity studies, Section 5, and matching the duration based on Table 1.
This section really defines the flow of a program. For very short clinical trials, say up to 2 weeks, you need a minimum 2-week study in 2 species. One of them has to be a non-rodent.
And there's a U.S. alternative for single-dose trials, isn't there?
There is. The U.S. regulatory landscape offers an alternative where extended single-dose tox studies can sometimes provide enough assurance.
And what happens when we're moving into really chronic dosing, clinical trials that are longer than 6 months?
Then the requirements solidify. You're looking at 6 months in rodents and 9 months in non-rodents as the standard. It's worth pointing out that European regulators generally consider 6 months in non-rodents to be acceptable. The principle is just that the animal exposure has to adequately capture the potential for cumulative toxicity.
Here's where it gets really interesting, the logistics of it. I'm thinking of footnote B in Section 5.1. It's a huge efficiency measure.
Oh, absolutely. Footnote B is a lifeline for so many programs. It lets you initiate clinical trials that run longer than 3 months.
Which is a lot of phase 2 trials.
A lot of them.
Yeah.
You can get them started with only 3-month tox data in both species. This can save months of clinical delay. But, and it's a big but, the final chronic study data, that 6- and 9-month data, has to be generated and available before you extend human dosing past that 3-month mark.
So it's a phased data submission. You get a sort of conditional approval to proceed as long as the full data follows right behind.
That's a good way to put it. And for serious or life-threatening indications, there's even more flexibility. You can proceed with just the complete chronic rodent data and, say, the in-life and necropsy data for the non-rodent, as long as the full histopathology report follows within another 3 months. It's a very structured risk tolerance.
Are there any unique clinical situations where you can justify shorter animal studies, even if the drug might be used for years?
Yes, the guideline recognizes 2 main exceptions. The first is for intermittent treatment. Think drugs for migraine or erectile dysfunction, where exposure isn't continuous.
Makes sense.
The second is for indications where patient life expectancy is inherently short, like some advanced cancers. Chronic toxicity past that point just isn't a relevant concern.
And just for contrast, how do these durations change when we go from supporting a trial to the final marketing authorization in Table 2?
They get longer and more definitive, as you'd expect. For any treatment that will go beyond 3 months in general practice, you absolutely must have the full 6-month rodent and 9-month non-rodent data. The rigor is required because the population at risk post-approval is just so much larger.
And less controlled than in a clinical trial setting.
Much less controlled.
Let's pivot to those specialized pathways for early development. Exploratory clinical trials in Section 7. These are designed for that ultra-early human data PK, PD biomarkers with really limited exposure.
And critically, with no intent to determine clinical tolerability. The whole point of these pathways was an acknowledgement that a full 14-day tox study was often overkill for simple PK profiling.
But there's a trade-off.
A huge trade-off. You gain speed, but you accept really severe dose limitations.
So let's start with the most constrained of them, the microdose approaches. That's approaches 1 and 2. What are those dose ceilings?
Okay, so approach 1 is the most restrictive. The total cumulative dose is limited to 100 micrograms, no more. And it also can't exceed 1/100th of the NOAEL. The non-clinical package for that is minimal. A single-dose toxicitative study in 1 species with TK.
And what's interesting is what's not required.
Right. Genotoxicity studies are explicitly not recommended here, which just reflects how minimal the exposure risk is.
Okay, that is truly ultra-low exposure. Approach 2 gives you a little more flexibility.
A little more. Approach 2 allows a total cumulative dose up to 500 micrograms, but it has to be spread over a maximum of 5 administrations. And each individual dose still has to be 100 micrograms or less.
So a slightly bigger package needs a slightly longer study.
A 7-day repeated dose study in one species. You get early human data, but you have to live within those strict microgram limits.
Let's move to the multiple dose trials, approaches 4 and 5. Both of these support up to 14 days of dosing into the therapeutic range, but not for MTD. Approach 5, though, has this unique safety check.
Approach 5 is a very smart mechanism. It's for programs that want to move fast, but check for unexpected species differences. It requires a standard 2-week study in a rodent species, which sets your NOAEL.
But not for the non-rodent.
For the non-rodent, you don't need a full 2-week study. Instead, it's a small, confirmatory non-rodent study. We're talking only 3 animals per sex, dosed for at least 3 days.
And what's the dose they get in that tiny group?
They're dosed specifically at the systemic exposure that's equivalent to the rodent NOAEL. It's just a really efficient way to confirm that the exposure level deemed safe in rodents is also well tolerated in a more relevant species.
And what if it fails? What if you see toxicity in that non-rodent group at the rodent NOAEL exposure?
Then you stop. Clinical administration has to be deferred immediately. You're then required to go back and conduct a full standard non-rodent tox study before you can even think about going to the clinic. It's a built-in safety valve.
Let's shift our focus now to some of these key non-clinical hurdles that can become major inflection points, starting with toxicokinetics and the testing of human metabolites.
This is an area where M3R2 brings some essential clarity and can save a lot of needless work. The guideline sets a very precise 2-part trigger for when you actually have to characterize a human metabolite non-clinically.
And both conditions have to be met, right?
Both, simultaneously. First, the metabolite has to be observed at exposures greater than 10% of the total drug-related exposure in humans. And second, the metabolite has to be present at significantly greater levels in humans than what you saw in your animal tox studies.
That dual trigger is so key. You need both high relative exposure and that human-specific imbalance before you have to do more work.
Precisely. It stops you from chasing low-level metabolites that aren't systemically dominant. If that trigger is met, though, additional testing is usually required before you start phase 3.
Next up, genotoxicity, section down. The requirements here are often phased depending on how far you're going in the clinic.
They are. For a single-dose clinical trial, a single gene mutation assay, usually the Ames test is enough. But as soon as you move to multiple dose trials, the exposure goes up and you need an additional assessment.
For chromosomal damage.
For chromosomal damage, right. In a mammalian system, either in vitro or in vivo. And the full standard battery of genotox tests that has to be complete before you can start phase 2 trials.
Makes sense. You need to know it's not damaging DNA before exposing larger populations. Okay, finally, let's talk about the high-risk area of reproduction toxicity and including women of childbearing potential, or WOCBP. This is where ethics and risk management really intersect.
The guideline basically gives sponsors 2 paths. You can either characterize the inherent risk, which means conducting the developmental tox studies in animals, or if those aren't done, you have to rigorously limit the risk by using stringent precautions to prevent pregnancy.
And when we say stringent precautions, what's the regulatory definition of that?
It's very tightly defined. It means mandatory pregnancy testing, confirming menstrual periods, and using highly effective methods of contraception. And highly effective means methods that result in a failure rate of less than 1% per year.
So hormonal methods, IUDs?
IUDs, vasectomy for partners, that kind of thing.
But even with all these risks, WOCBP are allowed in early trials without the full developmental tox package. What's the thinking there?
The guideline acknowledges that sometimes you need WOCBP for clinical relevance or just for enrollment speed, so they can be included in early trials under these really intensive controls for short durations, like up to 2 weeks.
And there's a bigger flexibility point, too.
There is.
Yeah.
This is the one that allows up to 150 WOCBP to get treatment for up to 3 months if you have preliminary reprotoxic data from 2 species, just focused on fetal survival and morphology, and all those strict pregnancy precautions are in place. It's a calculated risk management decision.
So we've covered the core pillars here: defining doses, matching study durations, these special pathways for microdosing, and the triggers for testing metabolites and managing risk to WOCBP. The detail here is just a testament to that global push for a standard approach. So what does this all mean for us operationally?
I think for anyone navigating a development program, there are 4 key takeaways. First, your dose selection is driven by that 50x AUC margin. That's your scientific limit. The 1,000 mg/kg is the mass limit, and it can only be exceeded up to 2,000 in very specific high-mass clinical scenarios.
Okay, number 2.
Second, while your animal study duration usually matches the clinical duration, you have to remember that footnote B flexibility. You can start a longer trial with 3-month data, but you better have a clear plan to get that chronic data submitted before your patients pass that 3-month mark.
Right, and third.
Third, if you're using an exploratory or microdose approach, the benefits of that streamlined package are tied directly to the strict microgram dose ceilings. You cannot violate those limits, or the entire justification for the expedited package is gone.
And finally, number 4.
Fourth, for metabolites, the trigger is unambiguous. It's 10% relative human exposure and significantly greater human levels. You have to use that precise trigger to avoid unnecessary work on irrelevant metabolites.
This guideline is really a living document, isn't it? It's always adapting, and it actively promotes the 3Rs: reduction, refinement, replacement, wherever it can.
It really does. And this leads to our final provocative thought. M3R2 was established, over a decade ago, and it mentions things like CERN-A as being unique cases. But given the absolute explosion in innovation gene therapies, mRNA platforms, sophisticated in silico toxicology, how quickly will the scientific community and the regulators validate these new non-animal methods to a point where they can truly replace standard toxicity testing? And would that necessitate a fundamental overhaul of these standards in the very near future?
That shift from the animal model to the computational model, that's definitely the horizon we're all watching. Thank you for joining us for this deep dive into the blueprint of non-clinical safety. We hope this helps you navigate your next critical development phase with confidence.